All right, so in the last 10 or 15 minutes, I want to cover part of the citric acid cycle. Obviously, the citric acid cycle contains, you should know one thing that's in the citric acid cycle, and what do you suppose that is?
Citric acid.
Citric acid. So, which technically... cycle. Yeah. That kind of thing. Technically, it's citrate in vivo. So it's already kind of, yeah. But, and it's also called the tricarboxylic acid cycle, or the TCA cycles. Why do you suppose it's called that? The tricarboxylic acid cycle.
There might be 3 carboxylic acids involved.
Well, it's because many of these, including citrate, has 3 carboxylic acids on it. And then it's also called Krebs cycle, or originally it was part of the Krebs bicycle. And so, all right.
Pronouncing it
Yeah.
sickle?
Okay. So for aerobic, first of all, if I've asked, I've had questions before, like kind of true, false, or whatever. The fact is, the citric acid cycle is an aerobic metabolic pathway. So if you're anaerobic, you're not doing the citric acid cycle. Okay, right? That's like lactate. You go down to lactate. So there are 3 processes in the aerobic portion of the metabolic pathways, and it's the citric acid cycle, which has 8 steps. 9 if you include what I call step 0. Some books put it in there. Some books don't. Yours doesn't, because it's technically in a different location. And it'll make more sense once you see it. So the citric acid cycle, there's sometimes they call it the ETC, the electron transport chain, which, if we go back to our little, so let's see if I can get this to work. Whiteboard. There really is a delay. Okay, so if we go back to our cell that we've been drawing, sometimes when we do these cells, I don't know why it comes up yellow. This is not yellow. I don't know if you can see it, but I think it's green. And so I don't know why it's coming up yellow. But sometimes we're talking specifically about the hepatocytes, because the liver does so much compared to certain other parts of your body. So we have the cytoplasm, and this is not drawn to scale. Then we'll talk about times when we've got the mitochondria. Okay. Like I said, definitely not drawn to scale. Here, let me see if I can just change.
You mean our cells aren't that large?
Well, that and the mitochondria is not that big. All right. All right, and so where does glycolysis occur?
Cytosol.
That's in the cytosol. So right here, glyco. Okay, at least the black writing is blacked on there. Okay, so that's glycolysis. Where does the citric acid cycle occur? So, you go through, if you're aerobic, which hopefully we are all breathing oxygen right now, if you go through glycolysis, you get to pyruvate. Okay, of course, we need to go on to the citric acid cycle next. Where is the citric acid cycle? Not quite, not in the inner membrane. There's one step that's in the inner membrane. The other 7 steps are elsewhere. Pardon?
The outer.
No.
Between the inner and the outer.
No. You need... Not the Christie. The matrix. So it's in here. Citric acid cycle, with one exception. There's one exception, there's one enzyme that is a membrane-bound protein that's in the inner membrane, okay, which is important because it's one that interacts with the electron transport chain. And where does the electron transport chain occur? In the what? In the inner membrane, okay, and so, which I'm just going to draw it in like this. Okay, so here's the electron transport chain, yeah. Later on, I'll draw it. I'll blow it up and show what it says. Now, the one exception for that is that cytochrome C is a transient one. It's in the inner membrane space in between the two, and it jumps, and so we'll see that. And then we've already talked about gluconeogenesis. It's a little tricky because remember, gluconeogenesis occurs. Well, it depends on where you start, but it's in both the mitochondrial matrix and in the cytosol. And so that's why it's going... Oh, sorry, we got kicked off. And now I'm trying to reconnect. Oh, come on now. Let me pause this. Okay. All right. And so it's really important to keep track of where everything is occurring. And then the third process is the fact that you have to do what's called the OXPHOS, oxidation or oxidative phosphorylation pathway, which is coupled to the ETC, the electron transport chain. And we have a whole chapter on that later on. And it's the reason being that the final electron acceptor is oxygen, that's why it's aerobic, and you ultimately make ATP. And it looks just like a little turnstile. It's really cool. Okay, and in general, if we're going to do a review, we have the metabolism consists of catabolic or catabolism and anabolism or anabolic parts. And so what is catabolism? You're breaking it down, and anabolism, you're building it up. And so is glycolysis a catabolic or an anabolic process? It's catabolic. Okay, whoa. Whereas gluconeogenesis would be anabolic. Okay, whoops. And for whatever reason, it didn't come through. But the citric acid cycle occurs in the mitochondrial matrix with the one exception that I'll point out. And it has to do with fumarase. And then the citric acid cycle is actually amphibolic because it plays a role in both catabolism and anabolism. Okay, and so we're going to see how citrate gets broken down and then ultimately you get it reformed. So it truly is cyclical, whereas glycolysis, gluconeogenesis were linear pathways. We've seen the pentose phosphate pathway, which I don't even know what shape you'd call that one, lattice, crisscross, or whatever. And then once we get to the fatty acid ones, you'll see that those are spiral. Okay.
Wait, so does OXPHOS also occur on the membrane?
It's on the inner membrane, yes. Yes. Okay. All right, and so I really like this picture. This one came out of the old book, but it just shows you the different stages in how the citric acid cycle, which I abbreviate here as CAC, relates to the catabolic pathways. And so we can see here, these are the three biggie catabolic pathways, and then it all comes down to a common metabolite, which is acetyl-CoA. Okay, so we're gonna find out how pyruvate becomes acetyl-CoA, because once again, pyruvate is sort of like Moses in the Holy Land. It can see that Holy Land of the citric acid cycle, but it cannot enter in. Faith integration. Amino acids, some of them you can break down to acetyl-CoA, and then fatty acids break down directly to acetyl-CoA. And so it's the acetyl-CoA that actually goes into that second stage, which would be the citric acid cycle. And then the whole purpose really is to create those reduced electron carriers, NADH, FADH2, those, and NADPH to a lesser extent for some of the other pathways. Those are the whole purpose, and it's because those are what participate in the electron transport chain to ultimately make ATP. Okay, so that's why that's really important. It's not, you don't get hardly any ATP directly from glycolysis. You only get a net gain of 2. And so we would be really sick and lethargic and would die probably if we could only rely on that. We get a lot from fatty acids, but oh, whoops, I did it again. We get a lot from the fatty acids in the sense that it just breaks down quickly to acetyl-CoA, which can ultimately go through the second stage. Okay.
So the electrons coming off of pyruvate going, like skipping stage 2, where are they?
So, no. Whenever they show this as being an electron, remember you made an NADH there.
Oh.
So that's just showing you that it's an NADH. It's just easier for it to draw as an E-. So each step, remember earlier in step was it 6 or whatever, we made an NADH. Technically, we made 2 of them from glycolysis, and so it can go on. And then we'll find out that, the fatty acids, amino acids, and stuff go on as well. So it's NADH or FADH2 that are the important parts there. This is the figure that was in your book, if you like it vertical, so be it. This is the citric acid cycle, okay? And so I'm going to quickly, because it's got all the names and stuff in there, but I want us to be able to use our logic and figure out what the names are. First up is what I call step 0. So technically speaking, it is not a part of the circle. So if that one really is dead. It's a little short, it doesn't really matter. But technically speaking, if the cycle really looks like this, and this is the part that's the cycle, then this is step 0. Okay. Because it's how you get the pyruvate. Remember, it's like Moses in the Holy Land. The pyruvate cannot enter into the citric acid cycle, and so it has to be modified first. This is called oxidative decarboxylation. Okay, and it's done by a major complex. And we're going to find out in chapter 20 that every NADH that you make, you get 2.5 ATPs. Whereas if you use FADH2, you only get 1.5. And between this chapter and chapter 20, it'll make sense the reason why there's a difference. So this complex actually has several cofactors along with it. So one is coenzyme A, because that's the CoA that becomes the acetyl-CoA. Another one's NAD plus becoming NADH. So you're going to make a thioester. So what class or classes of enzymes utilize NAD, NADH? Dehydrogenase. So if you're going to name this, what would you call it? This is pyruvate dehydrogenase complex, and that's because it is huge. I mean, this thing is massive. We're going to see another dehydrogenase complex that's very analogous to this later on. Okay. And so you do kick off a carbon dioxide, but you get the acetyl group onto the coenzyme A, which is the important part, because acetyl-CoA then can enter into that holy of holies and also known as the citric acid cycle.
So, do dehydrogenases always remove CO2?
No.
Just
Sometimes, yeah, because it can have multiple activities. Okay, an enzyme can't. It's not like an enzyme just has a one-track mind. Some enzymes can do more than one thing. Okay. This is very much an oversimplification, but it was what the old book shows. But it's this little cartoony way of showing the exact same thing. This is much better. Remember I said that there are 5. So there's coenzyme A, NAD, NADH, FAD, lipoic acid or lipoate, and TPP, thiamine pyrophosphate. Because, like I said, you can usually visualize these on an electron micrograph. That's how big this complex is. To me, it kind of looks like a honeycomb cereal. I don't know why, but it's what it reminds me of. Okay.
so that E1 plus E2 plus E3, there's 3 enzymes involved.
But they are physically attached to each other. It's a factory. That's why it's a complex versus just being a single enzyme. Like the quaternary structure is huge. I don't remember. I didn't print this one out so I don't remember if it's... Okay. So at this point, I'm going to go ahead and end because we're going to talk about each of these and what roles that they have and go in. Because like I said, we're going to see... A lot of times I abbreviate this one PDH just because it gets to be a pain to write out pyruvate dehydrogenase complex. And so we'll see what each of these players are. Okay? So go ahead and end class with prayer.